fourth and last of my videos in a series in which I attempt to introduce the biology of cancer. This one is about drivers, as we increasingly call them, that is, the mutations that drive cancer. In previous videos, I've discussed how mutations accumulate in cells to give rise to malignant cancers, using this diagram here. And I made the point that when I say mutations, I mean in the broadest sense, that is, anything that can happen to DNA, right up to the scale of whole chromosome changes. And we also must include epigenetic changes and things like viruses and mobile elements that alter genes. So it's really gene changes that drive cancers. So now I want to ask questions like, which genes get mutated, how many mutations are mutated in a typical cancer, and what sort of mutations occur? In other words, what sort of changes to the DNA or the epigenetic regulators occur? Let's start with which genes are altered in cancer. And the basic statement here is that all sorts of different genes and all sorts of different pathways and all sorts of different systems get modified. It's quite bewildering, really. I have a list here of all the different pathways and gene types that can get mutated in cancer. And it's not even a complete list, but I want to talk through it just to give you some idea of the enormous range of genes and systems that are involved. Starting at the top of our list, we have the mitogenic signaling pathways, and these are mutated in the model of colon cancer that we discussed in previous video. So, for example, we have the Wnt signaling pathway, the receptor tyrosine kinase pathways, both of which are represented as mutations that commonly occur in colon cancer. We also have the control of the cell cycle by the retinoblastoma protein RB, which I've also shown you in a previous video. And then we have a whole series of other mutations in pathways like the hedgehog pathway, the notch pathway, the hippo pathway. These are signaling pathways which control cell behavior, and their names sound a bit odd, but they're derived from mutations that were originally discovered in fruit flies in Drosophila, where they tend to give their gene names somewhat exotic names like hippo and hedgehog. So those are positive growth signaling systems on the whole. These are signaling systems that tend to encourage mitosis, but they're also inhibitory signaling pathways such as the TGF-beta signaling pathway, which again is mutated in the Vogelstein model of colon cancer that we've discussed previously. Then there are a whole series of mutations which occur in genes which control transcription, the production of messenger RNA. There are transcription factors themselves which control gene expression directly, and these include molecules like p53, which we've discussed before, beta-catenin, MYC, and members of the ETS families, which are all prominent in human cancer, and hormone receptors such as the estrogen receptor. These are hormone receptors that are in the nucleus and bind directly to DNA and control gene expression. Then transcription control systems also include the epigenetic modifiers which control the structure of chromatin. For example, the histone modifiers which add small groups like acetyl and methyl to histones and thereby regulate gene expression. What we haven't talked about before is the next one on my list, which is cell adhesion molecules. The cadherins are molecules which bind epithelial cells to each other at the junction at the top of an epithelial cell, viewed in a diagram like I've got here. The cadherins are cell-cell adhesion molecules, so the same protein is present on both cells and bind the two cells together to form an adherens junction, And among of the interesting things about the cadherins is that they interact strongly with beta-catenins, which are involved in regulation of mitosis, amongst other things, and are mutated in colon cancers. Moving on from cell adhesion, I should remind you that we've talked previously about telomerase. This is the enzyme that replicates telomeres at the ends of chromosomes. These little fluorescent marks in this diagram show the telomeres at the ends of chromosomes. These shorten as cells replicate. Telomerase is the enzyme that adds telomeres and so allows cells to go on dividing beyond their normal division limit, as we discussed in a previous video. We now know, for example, that the telomerase gene is turned on by rearrangements of DNA in quite a few human cancers. Then next on the list is the various DNA repair enzymes and proteins involved in mitotic activity, which are mutated to give rise to genetic instability. Finally, on our list, we have carbohydrate metabolism. It was no surprise when people discovered that there were mutations in carbohydrate metabolism in cancers. What was a surprise was that these mutations did something that was completely unexpected. I can't resist the temptation to go through this because it's one of the weirdest examples of mutations in cancer and shows you just how weird and how varied and how wonderful the mutations are. What we had expected was to find mutations in cancer cells that might bring on the Warburg effect, the fact that cancer cells tend to switch from oxidative phosphorylation to glycolysis as a source of energy. What was unexpected is that mutations which we find are quite different from this, and the best known are the mutations in the isocitrate dehydrogenases, IDH1 and IDH2. These are mutated in gliomas and leukemias, amongst other cancers. The isocitrate dehydrogenases are in the citric acid cycle, and they don't stop the citric acid cycle from working, but they change the specificity of the isocitrate dehydrogenase so that they divert some of the activity of the citric acid cycle into producing a molecule that would not normally be produced by the citric acid cycle. Instead of dehydrogenating isocitrate to alpha-ketoglutarate, these mutant forms of IDH produce hydroxyglutarate. And because hydroxyglutarate is not rapidly metabolized, it accumulates in cells and it interferes with other enzymic processes which are involved in demethylation. So the consequence of this mutation seems to be to block demethylation and therefore globally increase methylation in the cell. There are a number of possible consequences of this. The best understood at the moment are increased methylation of DNA and increased methylation of protein. The increased methylation of DNA may inhibit the transcription of certain genes. It's a kind of epigenetic instability mutation. And another possibility is that the methylation of proteins may also contribute to genetic instability by interfering with the DNA damage response. Whatever the consequences of this mutation are, they occur quite frequently in various human cancers, and clearly they make a difference. But they have nothing, as far as we can see, to do with the Warburg effect. Returning to our list, we've now run down a list of all sorts of weird and wonderful processes which are modified by mutations in cancer, and I've just ended my list by putting etc., etc., because I'm sure there are many other things that I haven't covered. The question of how many mutations it takes to make a human cancer has been addressed in various ways over the years. Mostly in the past, it was done by building mathematical models to try and explain how cancer incidence increased with age. And this led in particular to the estimate popularized by Armitage and Doll, that maybe there were 4 or 5 mutations in a typical adult human cancer. The trouble is that by building different mathematical models, you can come up with any number you like. And certainly Armitage and Doll admitted that their approach was very naive and probably not reliable. Nowadays, because we have access to sequencing data from cancers, we can address this more directly. And to understand how many mutations we think there are now, I need to spend a moment talking about the different kinds of mutation. So we need to distinguish mutations that occur at the sequence level, at the level of 1 or 2 base changes or a few base changes, which can either be a change in the bases or the insertion or deletion of bases, but are on a very small scale. And generally speaking, we set the scale limit at 50 or 100 base pairs. So mutations that affect less than 50 or 100 base pairs would be considered small mutations. At the other end of the spectrum, there are structural changes in chromosomes which can result in whole chromosomes being lost or gained, or bits of chromosomes being moved around, large-scale deletions, inversions, and so on. So I'll distinguish between small-scale changes and large-scale changes. And I'm going to focus particularly on the really small-scale changes, the individual base pair changes, because these are the ones we can analyze most efficiently. If we think now about the process of cancer development that I've covered several times before, where we have a normal cell acquires a mutation which makes it slightly abnormal, and then among its progeny, sometime later, another cell acquires another mutation which makes the cell slightly more abnormal, and so on. And these are the mutations that actually drive cancer, and we believe in most cases give the cells a selective advantage. But at the same time that these mutations are occurring, lots of other mutations are occurring, because of course the vast majority of mutations don't give rise to cancer. They don't contribute to the cancer process. In fact, they will often be deleterious. Most of them will just be neutral. So alongside the so-called driver mutations, which do contribute to cancer, there will be many more mutations which we call passengers, which are irrelevant, but are nevertheless there. And when you sequence the DNA, you will find them in large numbers. And so we have an important problem of distinguishing between the driver mutations and the many more passenger mutations which don't contribute to the cancer process. So how we distinguish between passengers and drivers, we can't always, but in the cases of single base changes, there is quite a good way. To distinguish between the passengers and the drivers, we use the fact that the drivers will mostly show selection. And for point mutations, we can determine that by looking to see whether or not the mutation changes an amino acid. A mutation that doesn't change the amino acid will, in the vast majority of cases, have no effect, and therefore it will serve as a proxy, a measure of the number of random mutations that do nothing. If we look at this diagram from a very important paper by Martin Corena et al. From 2017, this diagram summarizes how we do this. A typical gene which has no role in the process of cancer development will have a series of random mutations in it, and the distribution of these mutations between mutations that cause amino acid change, no amino acid change, or actually stop in translation is predictable. And we can even adjust this prediction to account for the fact that the pattern of mutation may vary a little bit according to the type of carcinogen which is giving rise to the mutations. Driver mutations will have a different pattern. They will either show an increase in the number of mutations that change amino acids, because obviously in the vast majority of cases you need to change an amino acid to have any biological effect, or you might see an increase in the number of translational stop codons if you're looking for a driver mutation that blocks the activity of that gene. And so if we look at the proportion of mutations which change amino acids and the proportion of changes that cause stop, we get an idea that the gene is the target of selected mutations. By applying this analysis to a very large number of tumours, the authors came up with the following conclusions: that the number of point mutations in driver genes in the following cancers was quite variable. So in a cancer like testicular cancer, which occurs mainly in young adults, the evidence was that there was only about one on average point mutation per case. Whereas at the other end of the spectrum, a typical adult cancer, colorectal cancer, had somewhere between 8 and 13 mutations on average. Breast cancer and ovarian cancer come in somewhere in between with about 4 or 5 point mutations. And it's important to stress that we're only talking about point mutations here, and as we'll see later, the number of structural mutations in DNA is substantial and will add to these numbers. An important point that this analysis raised was that of these mutations, this estimated number of selected mutations, only about half of them were in cancer genes that were already known cancer genes. In other words, about half the mutations that were suggested by this analysis were in genes which so far had not yet been identified as cancer driver genes, most likely because they're rather infrequently mutated, and so when you look at them as individual genes, they are not obviously frequently mutated. And a similar point, which is also important, is that even in genes like p53, which are very frequently mutated in human cancer, a significant proportion of the mutations that you see are passenger mutations. They're random mutations, even in cancer driver genes. For example, in the case of melanoma, which has a very high mutation rate, 75% of the mutations in the known driver genes are just random passenger mutations. This is a very important point from the clinical application of genome sequencing, because it means that just because you find a base pair change in a gene in a particular cancer doesn't mean that that gene is functionally mutated in that cancer. And this is quite a technical problem for contemporary attempts to bring sequencing into the clinical world. Just to reiterate, this analysis only works for point mutations. We don't have a measure of the random background rate of mutations that are structural changes in the DNA. And so in cancers like breast cancer and ovarian cancer, for example, where there's a lot of DNA rearrangement, it's very difficult to identify genes that are targets of this rearrangement, because it's not easy to distinguish between rearrangements that are simply random breaks in the genome and rearrangements that are targeting a specific gene. This is made worse by the fact that there are regions in the genome which are particularly prone to rearrangement and breakage, the so-called fragile sites, where it seems that the nature of the DNA and its environment make it particularly vulnerable to breakage. We can now compare this analysis with a number of mutations that have actually been found in cancers. And I'm going to use the data that was published in 2020 by the International Cancer Genome Consortium, who surveyed a large number of human cancers to get a general idea of the numbers of mutations and the pattern of mutations in these cancers. And although this data is now getting a little bit old, it will serve our purpose quite well. Because of the problem of not being able to identify selected mutations compared to random mutations in structural changes in the DNA, they simplified by just taking genes that were already known to be driver genes, because they were mutated as well or something like that, and if they found a break in the gene, they would count that as a driver mutation. So here are some of the figures they came up with. The average number of drivers found to date in colon cancer, small scale changes about 7, of which most would be point mutations, large-scale changes in known driver genes, about 2.5 on average per cancer. Note that 7 of small-scale changes compares with about 10 predicted by the selection analysis. So it looks as though a fair proportion of the gene changes in colon cancer at the point mutation level have been identified, but there's still some to come, which agrees with the analysis of Martin Corona et al. Now look at breast and ovarian cancer. The interesting thing here is that the balance between point mutation and structural mutation seems to be quite different. These cancers seem to be driven much more by structural changes to DNA than by point mutations. So we know of 2 point mutations on average, which are found in breast and ovarian cancers, whereas 6 driver genes are rearranged in these cancers, and this will be an underestimate because of our inability to identify driver mutations modified by structural changes in general. At this point, I want to make a digression briefly, because this analysis of whether or not mutations are selected in cancer can be applied not only to look for mutations that are selected, but also to look for mutations that are selected against. In a gene where mutations that change the protein structure are selected against, we'll see a predominance of mutations that don't change the amino acid sequence, so-called synonymous mutations. Now, this is important in the context of an immune response to cancer, because people who believe that there is an immune response to cancers tend to invoke the idea that when proteins are changed by mutation, they will excite an immune response and they'll be selected against. In other words, that cancer mutations should be selected against where they change protein structure. And so if you have a gene which doesn't participate in cancer, you should find that mutations will be selected against if they change the amino acid sequence. So as part of this analysis, it was possible to look at proteins in general in the cell to see how many of them showed signs of mutations that changed protein structure being selected against. And they didn't really find any sign of that at all. So on average, there's half a coding mutation lost per tumour within statistical errors. This tells us that on the whole, in cancers, mutations that change proteins are not selected against in genes in general, and so that argues against there being an effective immune response to cancer, which concurs with what I said in the previous video. The next major question I want to address is what sort of mutations actually occur in practice in genes? How is the DNA changed? And the short answer to this question is that everything you can possibly imagine. But let's look at a variety of these things. If we look at small-scale changes, the commonest mutations that we detect are single-base changes. We've met them before, and they affect famous genes like RAS and P53 and so on. Slightly more interesting, perhaps, are small insertions or deletions, so-called indels, and these are more important than you might immediately think, because typically they change the reading frame of the sequence and cause a frame shift. If you cause a frame shift in the translation of a protein, then generally speaking, you end up with a truncated protein because you run into stop codons. And I'll illustrate this with an important example of the mutations of the APC gene, which frequently involve indels and frame shifts. APC is the first gene mutated in the standard model of colon cancer, as shown here. About 80% or more of colon cancers have a mutation in APC, and it's very often a truncating mutation. Here's a real example from a real tumour. There's the amino acid sequence in green written underneath the DNA sequence, CCA, ATA, and so on, in purple. In this particular case, a T has been inserted next to an A, and this results in a shift in the reading frame. And actually, in the next codon, we find a stop codon. And this is a particularly striking example, but it's not that improbable, because it turns out that in the wrong reading frames of proteins, the sequence is peppered with stop codons, probably to prevent the protein from being produced if a frame shift occurs on the ribosome. So a frame shift mutation, insertion or deletion of 1 or 2 or 4 or 5 base pairs usually results in a truncated protein. Moving on to large-scale changes, I'll run through the various kinds of large-scale changes because there are some comments to be made. Using the EGF receptor, EGFR, as an example, one of the commonest kinds of mutations is duplication. This is very often a tandem duplication, so that the 2 copies of the gene are in the same orientation, but you can get inverted duplications. If the process of duplication or other means of replicating bits of the DNA continues, you can end up with many copies of the gene, illustrated here, and this is now called an amplified gene. In cancer biology, we talk about amplification as a specific term, which specifically means many copies of the gene. It's not just increased expression, it's specifically many copies of the gene. And you find amplification of genes is quite an important mechanism of cancer, and you can easily end up with 50 or 200 copies of a gene, which results, of course, in increased expression. Then there are structural changes in chromosomes, of which the best known are the translocations. And there's 2 main kinds of translocation, the reciprocal translocation or the unbalanced translocation, illustrated here. In a reciprocal translocation, 2 bits of chromosome are swapped over between chromosomes, and this reciprocal rearrangement can be exactly perfect, so that the 2 bits add up to the 2 complete chromosomes, or there can be some gains or losses at the translocation breakpoints. But in many adult cancers, you get unbalanced translocations, which can either arise through the loss of one of the reciprocal pairs or by extra copies of bits of chromosome being joined together in some as yet unknown process. Chromosome translocations are famous, particularly in leukemias, because they can sometimes, but certainly not always, they can sometimes end up in 2 genes being joined together to produce a completely new gene, where the beginning of gene A is joined to the end of gene B. How do we study these mutations? Small-scale changes in the DNA, of course, are found by sequencing. In the case of large-scale changes, increasingly this is done by sequencing, but the problem with the contemporary sequencing is that most of the sequencing that's being done at present is short sequences, and of course if you take short sequences, you don't get a very good large-scale picture. So although we can approximately determine large-scale changes from short read sequencing, it's not completely reliable. The way in which you use sequencing to find structural changes is illustrated here. So the common kind of sequencing that's mostly done at the moment in 2025 is Illumina sequencing, where you sequence typically 150 base pair reads from each end of fragments of DNA. These fragments are often 250 to 500 base pairs in length. They can't be much longer than that. And so you cover effectively a few hundred base pairs. In the case of a chromosome translocation or any other kind of rearrangement, when you look at the sequences that you get, they will align to different places in the genome. You use the reference genome find the position of the sequences you have in the reference genome, and then look and see whether these should be adjacent to each other or not. This is a fairly effective way of finding something like a chromosome translocation, but it's technically difficult and it does generate a certain number of artifacts. More traditionally, large-scale mutations are studied using cytogenetics. That's where it all started. And so we'll have a brief digression about cytogenetics because it's still a very important part of clinical practice and our understanding of cancer. Cytogenetics is based on the fact that in metaphase of mitosis, chromosomes bunch up and you can spread them on a slide and examine them. And by classical techniques such as banding, you can see the structure of the chromosomes to a certain extent, probably representing patterns of relatively more or less active chromatin, which enable a skilled cytogeneticist to recognize different parts of different chromosomes. So here's a chromosome spread from a normal cell or an almost normal cell. These chromosomes will then be lined up to see whether or not they're all normal. And the karyotype I'm showing here, the chromosome pattern I'm showing here, is an example where there's one chromosome translocation giving rise to the so-called Philadelphia chromosome, the tiny chromosome marked by the arrow in the bottom right. This actually turns out to be an exchange of material between chromosomes 9 and chromosome 22, resulting in two genes getting joined together to form the famous BCR-ABL chromosome translocation we'll come back to in a minute. However, cytogenetics relies on a skilled person being able to recognize fragments of chromosome from their banding pattern, and this breaks down when you get to the typical adult human cancers. Here's an example of the karyotype of a breast cancer, almost straight from the patient, from a primary culture of a breast cancer, and you can see that the numbers of chromosomes are very changed. The structure of the chromosomes is all over the place, and there's a row of chromosomes at the bottom of the diagram where the cytogeneticist was unable to confidently identify any of the bits of chromosome present. The rearrangements are just too many and too complicated for the bits of chromosome to be identified by banding. So this led to the development of fluorescent in situ hybridization methods where you hybridize fluorescent DNA to the chromosomes to identify different fragments. Here, for example, is a spread of chromosomes, a metaphase, and here we've put on a probe for chromosome 12. So this is a cocktail of sequences from chromosome 12 labelled with red fluorescence, and it lights up the whole of an apparently normal chromosome 12. Also on this slide is a probe for the region around the NMYC gene. It's about 100 kb piece of DNA which has been hybridized to a chromosome 2 to show the presence of the NMYC gene. And what I'm not showing you is the bottom of this metaphase where there is a spectacular chromosome here which is made up of a chromosome 12 with the insertion of hundreds of copies of the region around NMYC in this great long green strip. This is a beautiful example of gene amplification. The region around NMYC has been multiplied into some hundreds of copies, resulting in very high expression of NMYC. And this is taken from a human lung tumour. Amplification can be seen this way, and of course you can also see structural changes in chromosomes such as translocations. Another common technique which is in clinical use is to look for amplifications and deletions by counting the number of copies of different parts of the genome. Here's a diagram taken from such a study on a glioblastoma. It uses an older technique of hybridization. Nowadays, what you would do would be to sequence the genome of this glioblastoma using small sequences and you count the number of sequences that map to each different part of the genome, and this gives you a very good estimate of the number of copies of each different bit of the genome. This was done by the older technique of hybridization, but it serves our purposes. So on the vertical axis here is the number of copies, and along the bottom is the whole genome. So chromosome 1 is over on the left, then chromosome 2 and so on, over to chromosome X on the right. And if we put in the number of copies, most of the genome is present in 2 copies. Part of the genome is present in only 1 copy and appears a little bit lower on the vertical axis. And there are even bits of the genome that are present in 0 copies. The reason that the X chromosome is present in only 1 copy is, of course, this is a male cell line being compared to a normal female. The region of the genome which is present in many different copies is an amplified region with lots of copies, and this is where the EGF receptor is. So this glioblastoma has many copies of the EGF receptor gene. It also has an extra copy of chromosome 7, which this amplification is on. You will also notice there's this small region of chromosome 9 where there are no copies of a short section of DNA, and this is where the gene variously called P16, CDKN2A or INK4A is. This is a cell cycle regulating protein, and deletion of this gene allows cells to pass through certain cell cycle blocks. So this is a good example of how you use counting the numbers of copies of different parts of the genome to find deletions and amplifications. I now want to consider what mutations do to genes and the proteins they encode. And I'm chosen as an example the receptor tyrosine kinases and the pathways that take signals from the receptor tyrosine kinases to the nucleus, with EGF receptor shown here as a particularly good example. It's a transmembrane protein which binds on the outside of the cell protein ligands like EGF, and on the inside of the cell in the cytoplasm it phosphorylates itself, and the phosphorylated residues bind to other signaling proteins which link into signaling pathways. The 2 major signaling pathways I've shown here are the RAS-RAF to MAP kinase pathway and the PI3 kinase to AKT signaling pathway below. As well as the EGF receptor, there's its close relative called ErbB2 or HER2, which is famous in breast cancer as the target for the drug Herceptin. Let's consider some of the point mutations that occur in the components of these pathways. 3 of them are particularly famous for being point mutated: RAS, RAF, and PI3 kinase catalytic subunit, PI3KCA. These mutations are present in the Vogelstein model of colon cancer, which I popped in underneath. One of the mutation stages in the Vogelstein scheme is KRAS or BRAF being mutated, and another step is PI3 kinase or P10 being mutated. RAS is perhaps the best known of all these point mutated proteins. RAS is a GTPase which is activated by binding GTP, and when in the GTP bound form it's active in signaling to be RAF, and it has a GTPase activity built into it which hydrolyzes the GTP to GDP, and in that form it's inactive. So the signal that would be passed on down the pathway is inactivated by hydrolysis of GTP and activated by binding of fresh GTP. The inactivation is catalyzed, as well as the enzymic activity of RAS, by the binding of a GTPase activating protein, a GAP protein. The point mutations in RAS tend to inactivate the GTPase activity, and certainly some of them prevent the binding of the GTPase activating protein. which has the effect of reducing the rate at which GTP is hydrolyzed or preventing it altogether, and so the RAS remains in a permanently active signalling form. Its next neighbour down the pathway, BRAF, also undergoes point mutation. A diagram here shows how the commonest kind of mutation in BRAF is the change between valine 600 to a glutamic acid. And it's believed that what this does is it puts a negative charge into the protein chain in the amino acid next to threonine 599, which can be phosphorylated. It seems that threonine gets phosphorylated when RAF is to activate RAF, and so by putting a negative charge on the adjacent amino acid, this mimics the phosphorylation and at least partially activates the protein as though it had been phosphorylated. Among the other point mutations that occur, both the EGF receptor and ERB2 are activated by point mutations. P10 can be inactivated because it's a tumour suppressor gene. It can be inactivated by point mutation, and AKT downstream can also be activated by point mutation. Thinking about other kinds of mutation, the structural mutations, deletion is a good example, and P10 will suffer deletions. It's a gene that has to be inactivated in cancer, and so deleting it altogether, or part of the gene, is a common kind of mutation. Duplication is another example of a way of activating a gene, and this occurs in the case of BRAF. Zooming in on the RAF gene, BRAF is adjacent to another gene called KIAA1549, and if a piece of about 2 megabases spanning those two genes is duplicated, you end up with the structure shown here, where in the middle the KIA gene is joined to BRAF, and this fused gene, which makes a protein which is part KIA and part BRAF, that protein is an activated form of RAF. This is found particularly in astrocytomas, a kind of brain tumour. One of the take-home messages here is that the same gene can be activated by completely different mechanisms. In the case of BRAF, you can have either point mutation or you can have a tandem duplication activating it. And in fact, this applies to the other genes as well. So RAS can be activated by translocation, chromosome translocation in some cases, for example, although the point mutations are better known. As an example of amplification, we can go to the two tyrosine kinase receptors. The EGF receptor is often amplified in brain tumours of a particular kind, and ERB2 or HER2 is amplified in about 1 in 7 breast cancers. Finally, we come to chromosome translocation. As I've already mentioned, chromosome translocation can activate even a gene like RAS, but it's more famous for occurring in leukemias. The first human cancer mutation, in fact, was the Philadelphia chromosome translocation to form the Philadelphia chromosome, as I showed a few slides back. This is a reciprocal translocation between chromosomes 9 and 22, and it fuses the gene BCR to ABL. And to give you some idea of just how powerful these fusion gene effects can be, here's a diagram of the ABL gene. It's a tyrosine kinase, and it's controlled by its N-terminal domain. The N-terminal domain folds over and blocks the ABL kinase in the normal resting state. Side by side with that, I've put BCR, and the normal BCR likes to form dimers or possibly multimers. And when these two proteins are joined together by chromosome translocation, you have the front end of BCR joined to the tail end of ABL. And this has several effects. This gene will now be driven by the BCR promoter, for example, which may well alter the expression compared to Abl kinase. The BCR binds the protein together into dimers or multimers, and the Abl kinase has both lost its N-terminal controlling domain and also, by forcing it to be dimerized, it's activated, because tyrosine kinases are activated by dimerization, and when they're dimerized, they autophosphorylate. So this fusion protein is a superactivated form of Abl kinase, and it illustrates just how powerful and unexpected the effects of a chromosome translocation can be. The chromosome translocations were found and made famous in leukemias, and for a long time it appeared as though they were something that happened in leukemias, but the possibility that they occurred in the common adult cancers, epithelial cancers, was largely ignored, although there are one or two examples like thyroid cancer where chromosome translocations have been described. And then in 2005, it was shown that there was a very common translocation, or actually a deletion, causing a fusion gene in a common epithelial cancer in prostate cancer. This rearrangement brings the TMPRSS2 gene adjacent to the ERG gene by an internal deletion or other rearrangement on chromosome 21, and this fusion gene is present in about half of all prostate cancers. And because prostate cancers are the commonest human cancer of all, probably, this is clearly the commonest known human fusion gene. There are now many more examples of fusion genes in common adult cancers, although none are quite as abundant, as far as I know, as the TMPRSS2 ERG one. But for example, in lung cancer, you have the EML4 ALK fusion, and there are various fusions of the NRG1 gene known. An important point that comes out of this is that it's not just chromosome translocations that create fusion genes. Any kind of DNA rearrangement can create a fusion, so translocation, deletion, inversion, tandem duplication are all known to be able to give rise to fusion genes. It's obvious, really. But it's also worth remembering that more often rearrangements of the DNA will inactivate a gene rather than create a fusion. The vast majority of DNA rearrangements that break a gene will cause it to be inactivated, and of course quite a lot of rearrangements simply result in gene loss. As well as changes to the DNA as such, there are several other kinds of gene change that we mustn't forget. There are epigenetic changes, there are viruses, and there are transposons. Epigenetics first. There are several genes known to frequently be turned off by DNA methylation. It's a good example of an epigenetic change. MLH1 is a mismatched DNA repair enzyme, which is quite often silenced by methylation of its promoter in colorectal cancer. And cadherin E, which I mentioned earlier, is the cell-cell adhesion molecule that binds epithelial cell surfaces together. That is turned off in many lobular breast cancers by methylation of the DNA. Viruses. Several viruses encode proteins that block genes like P53 and RB, including the human papillomaviruses, in which case the proteins E6 and E7 are involved. The E7 protein binds to the retinoblastoma protein, and the E6 protein binds to P53. And the combination of blocking RB and P53 leads to freeing up of the cell cycle. Finally, there's the line 1 retrotransposons, which in human cancers are the most important kind of mobile element or transposon. They copy themselves into new places in the genome, and in some tumours, line 1 retrotransposons form 10 to 1,000 new insertions, some of which will create DNA rearrangements, some will interrupt genes, some may turn genes on. We don't have very many examples yet, but they clearly can be significant, and there are a couple of cases known where a line 1 retrotransposon has mutated the APC gene in colon cancer, for example. So in conclusion, we've seen that most adult human cancers will typically have, on average, 10 or more gene changes. These gene changes are in a bewildering variety of genes which can code for proteins which take part in a wide range of cellular functions. The mutations themselves can be small mutations like single base changes or small insertions or deletions, up to large mutations on any scale, including gain or loss of whole chromosomes. But in addition to this, there's epigenetic change. Viruses bring new genes into the cell, and mobile elements move around in the genome and disturb genes. So the bottom line is that cancer development is a very complicated process involving a bewildering array of genes and a bewildering array of mutations. That's the end of the fourth and the last of my videos on cancer biology. I hope you found them useful.